agarose, which is extracted from seaweed, or polyacrylamide. Here is a scanning electron micrograph of agarose. And you can see that this type of gel contains all kinds of barriers and pores, and it's used to separate DNA molecules. So a long DNA molecule traveling through this gel would bump into an obstruction, for example, and then the front end might turn into a blob. And then with the continued application of the electric field, the DNA could begin snaking through the gel again. So as a result, the DNA molecule would move in a discontinuous way through the gel, being retarded by the sieving properties of the agarose. And the agarose would separate the DNA on the basis of size. Now, here is a slab of agarose. The slab of agarose contains at the top some wells which allow you to load the DNA samples. So I'm going to load the DNA samples into the top of the agarose. And then I'm going to hook the gel up to an electric field. And because the DNA is generally negatively charged, I'm going to hook up the electrodes so that the positive charge is on the bottom. And as a result, the DNA will migrate down the gel and separate according to size. Now, what about separating biological molecules? You can separate either RNA, DNA, or protein. The DNA, as we mentioned before, is generally negatively charged because it's got phosphate groups in its phosphodiester backbone. The DNA is very long, so we cut it with a restriction enzyme. Okay. Then we have different sized DNA molecules. The restriction enzymes cut at very specific sequences. So the DNA molecules end up cut into very characteristic set of sizes. Then we separate the DNA molecules according to size by gel electrophoresis. We blot the DNA molecules onto a solid support, say nitrocellulose. And then, now that the DNA is on nitrocellulose, we can denature the DNA and then hybridize the denatured DNA to a p32 labeled DNA probe. The DNA probe will contain DNA sequences that correspond to the gene we're particularly interested in studying. This clever procedure for analyzing complex mixtures of DNA is called the Southern blot, named after the inventor of the method, Ed Southern. What about RNA? Well, RNA is single-stranded, and it often contains sequences that are self-complementary and therefore can form secondary structures. The RNA needs to be denatured prior to electrophoresis. And in the denaturation process, we end up with an extended piece of RNA, again negatively charged because the RNA has a phosphodiester backbone. And just as in the case of DNA, we can separate the RNA molecules by gel electrophoresis. We can blot the samples to nitrocellulose. We do not need to denature because the RNA is already denatured. And then we can hybridize the RNA to a p32 labeled DNA probe to look for the particular RNA species we're interested in. Now, this is very similar to the analysis of DNA. So in an homage to Ed Southern, this procedure is called a northern blot. What about protein? Well, protein, in addition to being able to form blobs, often contains sulfhydryl bonds that hold the blobs into place. So the proteins need to be reduced in order to break the disulfide bonds and denatured with SDS, sodium dodecyl sulfate. the sodium dodecyl sulfate is negatively charged, and as a result, the protein ends up coated with a negative charge, just as in the case of RNA and DNA. Just as before, one can do gel electrophoresis of the different proteins. One can blot the proteins over to nitrocellulose. And then, in order to detect specific proteins, what we would do would be to bind the protein we're interested in to a specific antibody. And in a further homage to Ed Southern, this is called a Western blot. So, Western blots analyze protein, Northern blots analyze RNA, and Southern blots analyze DNA. What about Southern blot? Here's an example. This is a Southern blot using probes derived from the fruit fly Drosophila melanogaster. These probes are for the UBX, FITC, and Antennapedia genes. These are homeotic genes that basically regulate the development of the body pattern of the animal. It turns out that these genes are strongly conserved in other animals, such as the beetle, and even conserved in the chicken and the mouse. The beauty of Southern blot is that the hybridization of the DNA probe doesn't have to be perfect. So the DNA probe will hybridize the sequences that are similar but not exactly complementary. As a result, you can find evidence for these genes in all these species. You can tell from the appearance of these Southern blots that the hybridization to the chicken and the mouse wasn't as strong because there's more background here between the bands. And that means that the stringency of the hybridization was decreased, and to bring out, to allow the hybridization to the specific bands, but as a result, there is a higher background. What about Northern blot? This is a Northern blot with 3 isoforms of PPAR, delta, alpha, and gamma. And it's analyzing RNA from various different tissues. Okay? And what these investigators are interested in are the RNA species expressed at different levels in different tissues? And you can see, indeed, they are. You can see, for example, that this isoform is expressed in most tissues, but not so much in liver. This isoform is strongly expressed in heart, kidney, liver, and BAT. And this isoform has its own specific expression in only 2 tissues. Now, we know that these changes across the board are real because there is a control lane containing a very abundant form of RNA, 18S ribosomal RNA. And this serves as a control that verifies that the same amount of total RNA was loaded into each lane. What about Western blot to analyze proteins? This is the analysis of the Wiskott-Aldrich syndrome protein in peripheral blood mononuclear cells. And this is an experiment which shows a healthy control and a patient with Wiskott-Aldrich syndrome. And patient number 1 here is being analyzed as a function of number of months after gene therapy. And the hope is that they could express the protein in the cells of the patient. And you can see at 7, 12, 15, 22, and 29 months, you can see that the level of the protein at 7 months was very low. At 12 months, it went up even higher. And then at 15 months, it went up higher yet. There's a hint that maybe the expression went down at 22 and 29 months. But if you look at the control, which is GAPDH, a housekeeping protein, you can see that perhaps that it looks like the amount of protein loaded in the lanes for 22 months and 29 months was somewhat less, so that the apparent decrease in expression at those time points is probably not real. So there you have it. That's our survey of gel electrophoresis of biological molecules. Thank you.